Dear students, Module 17, Translocations with suitable examples. The main objectives of this episode are to know about the chromosomal abnormalities and their consequences, to explain about the classification of chromosomal translocations, to understand the cause for the origin of translocations, to know about the balance and unbalanced translocations in human diseases, and also to explain about the translocations causing spontaneous abortions. Introduction. Structural chromosomal aberrations are the result of chromosomal breakage and the abnormal reunion of broken chromosomes. These can be produced experimentally by treating active cells to mutagens, namely as ionizing radiations, while spontaneous structural aberrations in both somatic and germ cells arise from the error of recombinations. Meiotic recombination preceded by synapsis of homologous chromosomes, and mismatching can occur in this process, particularly at chromosomal segments with tandem repeats of DNA sequences. It may result in duplication or deletions of DNA at such sites. Similarly, synapses between homologous sites on non-homologous chromosomes may lead to accidental recombination between non-homologous chromosomes. It results in the transfer of chromosomal segments from one chromosome to another. These rearrangements are referred to as translocations. Translocations serve as important diagnostic markers and therapeutic targets for leukemia, lymphomas, and many types of solid tumors. Rather than representing a random event, recent studies indicate that chromosomal translocation is a non-random event in a spatially regulated, site-specific, signal-driven manner and reflecting actions involved in transcription activation, epigenetic regulation, and DNA damages. In this model, the following aspects are emphasized: A. Classification of translocations. C. Balanced and unbalanced translocations. D. Role of translocation in diseases. And E. Chromosomal abnormalities in spontaneous abortions. Classification of translocations. Translocation is defined as the transfer of a part of one chromosome to non-homologous chromosome. When there is an exchange of segments on two non-homologous chromosomes, it is referred to as reciprocal translocation. It also includes the exchange of segments between non-homologous parts of a pair of chromosomes, that is X and Y chromosomes. The segment is neither lost nor added, but it is just exchanged. It was first discovered in Drosophila by the unusual behavior of a particular gene on chromosome 2 called PALI. It is lethal in homozygous condition. Bridges found that its lethality can be suppressed by the presence of another gene on chromosome 3, which is also lethal in homozygous condition. Stern in 1926 observed translocations of some allele on the Y chromosomes to the X chromosome. Translocation may be classified based on the translocated segment being present in the same homologous or non-homologous chromosome and number of breaks involved in the translocation. Translocations can be classified based on the involvement of the same or different chromosomes, intrachromosomal translocations and interchromosomal translocations. Intrachromosomal translocation is where the segment of a chromosome is shifted from its original position to some other position within the same chromosome. It is further subdivided into intraradial and extraradial translocation. In intraradial, the shift is occurring in the same arm. In extraradial, the shift occurs from one arm to the another arm. Interchromosomal translocation is where the segment is transferred from one chromosome to another one. It can be either fraternal or external. Fraternal is where the chromosome segment is translocated into the homologous. External is where the chromosome segment is translocated into a non-homologous chromosome. Classification based on the number of breaks involved: simple translocations, reciprocal translocations, shift or intercalary translocation, and complex translocation. Simple translocation involves one break and the terminal segment of chromosome integrated at the one end of the non-homologous chromosome. Painter and Muller in 1929 reported such type of translocations in Drosophila. Reciprocal translocation is where the segments are exchanged between two non-homologous chromosomes. It has one break in each of the involved chromosomes. Most of the translocations are reciprocal translocations and have been extensively studied in various plants and animal species. Shift translocation involves 3 breaks and the broken segment is shifted or transferred to the intercalary positions. Complex translocation is where more than 3 breaks are involved. Mostly, such translocations are naturally occurring. Translocation heterozygotes and formation of ring chromosome. Translocation heterozygote usually form the same number of homologous pairs as the normal homozygote, as well as centromere is not lost. But the result of pairing and meiosis are different in translocation heterozygote bearing 2 translocated segments and their normal counterpart. A reciprocal translocation forms a 4 chromosome complex at the Pachytene stage. The chiasma between such chromosomes may form a quadrivalent which can then disjoin in 3 different segregation patterns in the first meiotic division. Synopsis of heterozygous translocation chromosomes showing cross-like configuration later on opens out a ring structure. Consequences of such segregations are that of independent assortment between genes and non-homologous chromosome will be inhibited. Because of duplications and deficiencies, neither of the single mutant phenotypes will appear in the offspring. Translocation heterozygotes have low fertility. If the extent of duplication and deficiency is small, unbalanced gamete or zygote may not be lethal. Origin of translocations. Translocation may originate spontaneously. They may be induced by mutagens such as ionizing radiation and many chemical mutagens, since they induce chromosome breakage. Genetically controlled breakage in the chromosome may also produce translocations such as sticky gene and DSAC system in maize. In maize plant, semi-sterility was reported by Brink in 1927. In 1930, Burnham set a ring of 4 chromosomes in the semi-sterile plant of maize. In the same year, McClintock showed that translocation heterozygotes produced a cross-shaped configuration at Packetine. In Drosophila, the first translocation where a piece of the X chromosome was attached to the Y chromosome was reported by Stern in 1926. Specific genes have been reported to induce chromosome breaks, lead to the production of translocations. Genetically controlled system of chromosome breakage have been observed in some cases. Balanced and unbalanced translocations. The translocation can be balanced or unbalanced. In the case of balanced translocation, the chromosomes undergo rearrangements and no chromosomal gain or loss will take place. It can turn into unbalanced when the rearrangements inherits to the next generation. If neither parent carries a balanced translocation, their child may inherit an unbalanced translocation where there is an extra piece of one chromosome or missing piece of another chromosome. Frequently, a child can be born with a translocation, although both parents' chromosomes are normal. This is called de novo or new rearrangements. A child with an unbalanced translocation may have a learning disability, developmental delay, and other health problems. The severity of the disability depends on precisely the parts of the chromosome involved and parts of the missing or extra chromosome material. There are mainly 2 types of translocations. They are reciprocal and Robertsonian translocations. In the case of reciprocal, 2 detached fragments of 2 different chromosomes are estimates of incidence range are about 1 in 500 to 625 human newborns. This type of translocations are harmless and can be found through prenatal diagnosis, while carriers of balanced reciprocal translocations have increased risk of creating gametes with unbalanced chromosome translocations. Robertsonian translocation occurs when 2 non-homologous chromosomes get attached, were given 2 healthy pairs of chromosomes, one of each pair sticks together. It happens by breaks at or near the centromere of 2 acrocentric chromosomes, that is human chromosome 13, 14, 15, 21 and 22nd. Carriers of Robertsonian translocation seen in about 1 in 500 births. The reciprocal exchange of parts results in a large metacentric chromosome with 1 tiny chromosome, which can be lost from the organism with little effect because it contains few genes. role of translocation in disease. It is essential to distinguish between chromosomal translocation occurring in gametogenesis due to errors in meiosis and translocation that occur in the cellular division of somatic cells due to errors in mitosis. Somatic translocation result in abnormalities featured only in the affected cell line, as in chronic myelogenous leukemia, that is CML, with the Philadelphia chromosome translocations. Philadelphia chromosome is the first tumor-specific chromosomal change discovered by Nowell and Hungerford in 1960. Philadelphia translocation has a specific genetic abnormalities in chromosome 22 of leukemia cancer cells. The chromosome is defective and short due to the reciprocal translocation between chromosome 9 and 22, that is T 9 to 22, Q34 to Q11. This lead to the fusion of genes called BCR-ABL1. The ABL1 gene on chromosome 9 juxtaposed onto the breakpoints cluster region of the BCR gene on chromosome 22. It codes for a hybrid protein, tyrosine kinase signaling protein, that is always causes the cells to divide uncontrollably by interrupting the stability of the genome and impairing various signaling pathways governing the cell cycle. Chromosomal translocation between the sex chromosome may result in a number of genetic conditions such as XX male syndrome, where the translocation of the SRY gene from the Y chromosome to X chromosome takes place. Translocation in human can also cause infertility, and most common translocation in human involve chromosome 13 and 14 and is found in about 0.97 per 1000 newborns. The carriers of Robertsonian translocation occur in about 1 in 500 births. The common translocations are 45T, 13Q to 14Q and 45T, 14Q to 21Q, as these can lead to unbalanced offspring with translocation trisomy 13 and translocation trisomy 21 respectively. Chromosomal abnormalities in spontaneous abortions. Translocation may be completely harmless or they may cause serious health problems depending on the circumstances. Many people may have translocation without being aware of the conditions. This is usually the case for reciprocal translocation, a type of chromosomal translocation that increases the risk of recurrent miscarriages. Before the 20th week of pregnancy, a fetus is lost, which is called a miscarriage. More than 80% of miscarriages occur within the first 3 months of pregnancy. The causes of spontaneous abortion may be maternal, fetal, and placental, and chromosomal abnormalities may cause 50% of recurrent pregnancy loss. A person has all the genetic material necessary for normal growth in a balanced translocation. A chromosome merely broke and fused to another one. When an individual cells divide to create an egg or sperm cells for reproduction, these will end up with extra genetic material or missing genetic material, which could lead to miscarriages depending on which chromosome or genes are affected. In about 4.5% of all couples with recurrent miscarriages, one or both parents have a balanced translocation. Recurrent miscarriages may have physical consequences. For some women, repeated miscarriages can cause complications such as a buildup of scar tissue after a dilation and cutages, a routine surgical procedure performed on women following a miscarriage during the first trimester or to treat conditions that affect the uterus. Jumping translocation in spontaneous abortion. Chromosome translocation involving one donor chromosome and multiple recipient chromosome is referred to as jumping translocation. Acquired jumping translocations are commonly observed in cancer patients, mainly involving chromosome 1. Constitution form of jumping translocations mostly involve the acrocentric chromosomes and their satellites. It has been reported in patients with clinical abnormalities. Recognizable phenotype resulting from these events has included Down, Prader-Willi and DiGeorge syndromes. The breakpoints of all GTs occur in areas rich in repetitive DNA, that is telomeric, centromeric and NVARs. The unstable nature of GT resulting in chromosomal imbalance most likely contributed to the fetal loss. It appears that GT events may predispose to chromosomal imbalance via non-disjunction and chromosomal rearrangements and therefore may be unrecognized cause of fetal loss. Symptoms of spontaneous abortion include crampy pelvic pain, bleeding and eventually expulsion of tissue. Late spontaneous abortion may begin with a gush of fluid when the membranes ruptures. Hemorrhage is rarely massive. A dilated cervix indicates that abortion is unavoidable. To conclude, translocation show adverse effects on human health and play a role in spontaneous abortions. Proper genetic test and counseling are required for the family to prevent the chances of occurring genetic conditions. Thank you.